Welcome to this video. We are going to talk about the digestion and absorption of carbohydrate. Now, let's denote this as a piece of yam. The breakdown of this piece of yam into this simple units. The smallest unit, even more smaller than this, that the body is now able to absorb, that the GIT is able to absorb. That process, this whole process, is referred to as digestion of carbohydrate. And now, this breakdown requires enzyme. Carbohydrate digestion begins in the mouth. This is the mouth. Ptyalin is being secreted by the parotid gland. This ptyalin helps in the lubrication and solubility of this dietary carbohydrate that is being taken. And let's say a piece of yam that is being taken. This ptyalin helps in the lubrication. Now it begins in the mouth. There is no carbohydrate digestion in the stomach. Now the activity of the first phase of digestion of carbohydrates starts from the mouth, but it only lasted for some seconds, some few seconds. Some of us are even faster than that. The activity of this ptyalin continues down to the stomach. The optimum pH of ptyalin is 6.8. If ptyalin has a pH less than 4.0, it becomes inactive. Now this salivary amylase action continues down to the stomach. Because of the acidic nature, or due to the acidic nature of the stomach, the ptyalin, which is the salivary amylase or the alpha amylase, becomes inactive. Now, the second phase of carbohydrate digestion continues in the small intestine. This is a small intestine. Now, around the small intestine, there's what is referred to as the duodenum. Now, this partially hydrolyzed carbohydrate is also referred to as chyme. The physiologic student will understand that better. Now, upon the chymes entering into the small intestine, there are cells, the eye cells found in the mucosal lining of the duodenum. They release what we call cholecystokinin into the bloodstream. When this cholecystokinin is being released into the bloodstream, it goes to the pancreas. This is the pancreas. When it goes to the pancreas it stimulates the secretion of pancreatic amylase. This pancreatic amylase works in the same manner like that of the salivary amylase, but has more catalytic power than the salivary amylase. Now, this pancreatic amylase hydrolyzes maltose maltotriose and also starch. Maltose is 2 glucose units. 2 glucose units. Maltotriose is 3 glucose units. Now all of this is happening in the small intestine. In the small intestine, the absorption of carbohydrate takes place. Before then, what is referred to as enterocrine is being released. To stimulate, this enterocrine is released by the glands of Lubecun. We call it glands of Lubecun. It releases what we call enterocrine. This enterocrine stimulates the secretion of intestinal juice. Intestinal juice, which is also referred to as sulcus entericus. Sulcus entericus. Now this sulcus entericus contains enzymes like disaccharidase. Let's write it this way. It contains enzymes like disaccharidase, sucrase, lactase, and many others. If we have this to be a glucose structure, this is a carbon 6, the 5, the 4, the 3, the 2, and the 1. And we have the another glucose structure. We have this to be the 1, the 2, the 3, the 4, the 5, and the 6.
1,4-glycosidic.
The link between this carbon 1 and carbon 4 is called glycosidic linkage. Glycosidic linkage. You can also have a glucose unit linked from the carbon 1 to the carbon 6. This is a glucose carbon 1 to 6. That is a 1,6 glycosidic linkage.